Hello everyone, today I'm Srikan and along with my group mates are going to present you about mass spectrometry, especially with the focus on protein analysis and proteomics. And here is our outlines for our presentation. First, let's start with how the principle works. Mass spectrometry is a common technique to separate compounds in the samples based on their mass-to-charge ratio. It is often used in a couple with other techniques for protein identification. In principle, it involves three main components: an ionization source, a mass analyzer, and a detector. The procedures involve ionizing the sample, putting them into the electromagnetic mass analyzer to generate a result called mass spectra. All the three components are available with variety in techniques. Selecting specific ones and the realization of the protocols require consideration of the sample you are working with and measurement of the goal you want to achieve. But first, to understand its principle, let's look at the simplified models of mass spectrometry in action. Starting from injecting the sample into an ionization source, this sample needs to be turned into gaseous states and then ionized to give them electromagnetic reactivity. After that, the mass analyzer separates each ion's compounds by mass-to-charge ratio. For example, in our model, use a very basic analyzers where we first accelerate the ionized sample through a parallel electric field and then curve them with a normal magnetic field. The degree of curvature is determined by each ion mass-to-charge ratio, where under the same curve, where under the same charge, lighter ions curve more and the heavier ions curve less due to inertia. In our model, we can see that the radius of the curved paths are proportional to the square root of mass to charge. Of course, in actual MS are more complex, but the main principle of physics is the same. It is based on the motion of charged particle under the Lorentz force law, which results in the following ODE. For any analyzer, this core equation shows that under electromagnetic fields, charge to mass-to-charge ratio influenced ion dynamics through time. This observable difference caused by the analyzers, whether it is time of flight, path travel, energy or frequency differences, are detected by their respective detectors, where the signal is then processed into mass spectra by the data system. And in the next part, we will see how all these principles come into realization in actual application.
An essential beginning to the procedure is sample separation, as it can be seen in this figure. And for that, chromatography methods are applied along with mass spectrometry. Liquid chromatography combined with mass spectrometry has been becoming very preferable due to their combined resolving power and detection specificity. Separation process consists of mobile and stationary phases. The mobile phase, a gas or a fluid, flows through the stationary phase, which is a solid, and carries the sample with it. Different components end up traveling in different rates as a result of the introduction with stationary phase, so they become separated. As mass spectrometry measurements are based on mass-to-charge ratio, ionization is essential so that samples become either positively or negatively charged. There are three ionization techniques visible here that are mostly used. Electrospray is the most common one to be used for most type of compounds, and it's mainly dependent on polarity and molecular weight. Atmospheric pressure chemical ionization, APCI, could be necessary dependent on the compound, when electrospray method is not very preferable. And MALDI concerns the use of lasers striking a matrix of small molecules to make the analyzed molecules into the gas phase. After the ionization process, ions are sent to the mass analyzer where they are separated based on their charge-to-mass ratios. This separation can be done using electric and magnetic field or based on velocity of ions, like in time of flight, so TOF mass analyzer. Afterwards, outputs are sent to the detector, converted to a digital output. The last two sections are taking place in vacuum for most of the devices in order to prevent collisions with air particles. This diagram here shows the most common steps of sample preparation, but the protocol must be tailored to the specific samples for optimal results. Cell lysis is the first step in protein extraction. Lysate samples are prepared from biological specimens. In solution, digestion entails irreversibly breaking disulfide bonds via reduction and digestion of protein into peptide fragments. Another approach is to first resolve proteins by electrophoresis and then collect the gel slices that contain the desired bands. The protein in these gel plugs are then reduced and digested. After peptides are removed from the gel matrix, salt and detergents are removed as well, and they're prepared finally for the analysis.
And for the analysis of the results, DMS gives a result as a mass spectrum, which is essentially a histogram where the x-axis is the mass to charge value, and the y-axis shows the frequency, which indicates the relative abundance of the detected ions within the sample. These data represent a distribution of fragmented mass within the compounds of your sample, which in the case of protein analysis are amino acids and peptide fragments. The interpretation of mass spectra requires combined use of various techniques because one peak could indicate several biomolecular ions, and also one molecular ions can show up at multiple mass to charge value. Usually, for protein identification, the peptide mass fingerprinting is often used. The result is compared with the library of already known mass spectrum patterns, such as from the NIST-MS peptide spectrum database, with help of software such as Andromeda or Bionic for proteomic studies.
Now we will focus more on the limitations of the technique. When coupled to liquid chromatography, we saw that it is automated, multidimensional, and highly sensitive. However, at the same time, it's quite time-consuming and can be too sensitive towards the interfering compounds. And specifically, the mass spectrometer has limited dynamic range due to instrument detector saturation at high ion intensities. And pitfalls in peptide identification have been observed during sample preparations due to the procedure. And on multi-ionization, it was seen that there is weak identification of hydrophobic peptides, and it was inefficient in case of inhomogeneous distribution. And for electrospray ionization technique, it was observed that it was very sensitive to modest amounts of impurities.
Hi, so the next part I'm going to introduce you an example in which they use mass spectrometry to study the interaction between protein molecules. The name of the article is Identification and Characterization of Fibronectin Binding Peptide in Gelatin, written by Yu-Ying Liu, published on Polymer Journal in 2022. The first part, introduction, is to give you the context, general idea, and purpose of this research. Here I have the figure illustrating the extracellular matrix of the cells. This is a large network of proteins and other molecules that surround, support, and give structure to cells and tissue in the body. In this experiment, we have collagen. This is the main structural protein in the extracellular matrix, and it is found in various connective tissue of the body. The second object of the research is fibronectin, or FN. It's a glycoprotein and also a component of the ECM matrix. In the ECM, the conjugation of collagen and FN has important biological significance. The binding of collagen to FN could cause a conformation chain in FN, leading to various biological response, such as attachment to the cell surface to promote cell migration and wound repair, as well as difference in integrin receptor binding. Consequently, the study of collagen and FN binding sites is key to some relevant biological interaction. And how did they manage to study this important interaction? Well, the answer is with the help of biomolecular techniques, especially mass spectrometry or MS. So first we have bovine gelatin, which is the degraded form of collagen. The precipitated gelatin is recovered in trypsin solution. In this step, all the peptides in the gelatin will be digested, meaning being cut down to small peptide by enzyme trypsin. The mixture is then passed through Fn affinity chromatography, meaning only peptide molecule in the gelatin that can bind to Fn will stay, while other will be eluded. The molecular weight range of the sample is then analyzed by Maditov MS. To further investigate the amino acid sequence of the sample peptide, APLC MS is employed. So as you can see, by using information obtained by Maditov MS, which is the molecular range, the researcher can look at the effect of molecular weight to Fn collagen binding to see whether heavier peptides will bind stronger with Fn or not. Similarly, the information provided by HPLC-MS about the amino acid sequence of the peptide can also identify the specific binding site of Fn on collagen. Warning, you may see some really complex figures, but they are actually the spectrum result obtained from this article. And to not scare you with this very complicated diagram, I will summarize the result. What can mass spectrometry do from this spectrum? And what about other peptides? And what can we say about our peptides? The Maditov MS right here can confirm that the gelatin has been degraded and the fragment mainly distribute in the range of 2,000 to 3,000 Dalton. This result is the base to conduct further experiment combined with DPA analysis suggested that the peptide lower than 1 kilodalton did not readily bind to Fn. Thus, the bigger molecular weight of peptide, the better Fn binding activity. Therefore, the molecular weight plays an important role in interaction between Fn and peptide. The HPLC-MS result indicated that all Fn binding peptide contain GPAG or GPPG, or in other words, they are the sequence involved in the binding site of Fn on collagens. HPLC-MS also identified two peptides with similar sequence except for hydroxylation of proline, and these two sequences were synthesized. The Fn binding properties of synthesized peptide were determined by Maditov MS. For the first peptide, hydroxylation of pH and P15 is necessary for Fn binding. Thus, Fn binding properties are proline hydroxylation dependent. In conclusion, mass spectrometry plays an important role in the study of interaction between proteins.
However, the application of mass spectrometry is not limited to the identification of protein. With this technique, scientists can analyze big data such as proteomics. Let us illustrate this with an example from a paper titled "An Orthogonal Proteomic Survey Uncovers Novel Zika Virus Host Factors," published in the journal Nature in 2018. The purpose of the article is to identify cellular pathways that Zika virus use inside the host organism. Post-translational modification of proteins, especially phosphorylation, have crucial roles in controlling the complex reorganization of signal transaction pathways upon virus infections. The researchers therefore used liquid chromatography coupled with tandem mass spectrometry and demonstrated that among about 14,000 unique phosphorylation sites, more than 1,200 were altered. This approach allowed to map specific modification events along entire cellular pathways, highlighting modulation of key processes such as nervous system development, cellular assembly and organization, cell cycle regulation, etc. This information revealed several host factors that promote replication of Zika virus inside of the organism and that can be silenced to prevent the infection. So in this slide you can see the references that we used for our presentation. Thank you for the listening.